cats lay sleeping. Oh, yeah. Squatch-D TV, they're on the case. Steve and Chris, no mystery they can't face. Busting hoaxes, exposing lies. They're the champs of the Bigfoot mystery. Mystery. Bringing truth, science, and clarity. Squatch-D TV, they're the real deal. Uncovering the truth, it's time to reveal. Squatch-D TV, take us on your quest. Find the legend, put the rumors to rest. Late at night, shadows creeping. Deep in the woods, secrets lay sleeping. Oh yeah. Squatch-D TV, they're on the case. Steve and Chris, no mystery they can't face. Busting hoaxes, exposing lies. They're the champs of the Bigfoot mystery. Bringing truth, science, and clarity. Squatch-D TV, they're the real deal. Uncovering the truth, it's time to reveal.
Welcome to Squatch-D TV, where the truth is first before all else. Join your hosts, veteran investigator, TV personality, and author Steve Coles, and from the Kentucky Bigfoot Research Project and the Kentucky Fried Fixes channel, Chris Bennett. Sit back and prepare to take yourself on a journey with the longest-running Bigfoot podcast, Squatch-D TV. Here are your hosts, Steve and Chris.
And good evening, cyberspace. Welcome to Squatch-D TV for today's date, August 18th, 2024. I'm your host, your guide, Squatch Stick of Steve Coles, along with my co-host with the most, right down there, Mr. Chris Bennett. Hello, Chris. How are you?
Hey, Steve, man, it's good to see you. I'm glad to hear that neck is feeling better.
Yeah, it's doing pretty good today, yeah.
That's great, man.
Gots are working, so.
Well, what's the weather been like in New York? We've had some hot stuff here lately in Kentucky, and storms.
Rain, a lot of rain.
I don't know what we're getting the storm off of, but man, we got some plow winds yesterday that was really way up there. Did a lot of damage around here locally.
Well, we had, last week we had Hurricane Debbie make an appearance up here.
Yep.
And that was a very wet, very interesting day or two, but there was flood warnings, tornado warnings, and we avoided them all. So, and considering I only live about 200 yards from the Hudson River, that's not too bad.
Yeah. Well, that's kind of handy too. Go fishing.
Yeah, in fact, every day. In fact, even yesterday it was very overcast as I'm driving over the bridge to come home.
Yeah.
Because I have a narrow bridge that leads onto the state route, going towards like the interstate.
Yeah.
So it's very cool. When I come home, I have to go over this little trestle metal narrow bridge, and usually when I drive over, there's somebody in a boat with a fishing line in there fishing in the Hudson.
There you go.
So it's always cool to watch. Or there's some geese on the water, or because as you travel northbound into Greenwich, as you're going over the bridge to the right, where that water, it's a very swampy area of the Hudson shore. So you often see geese. Occasionally you'll see a crane over there, at least standing there in the water.
Well, yeah, I noticed where you live, there's a lot of wildlife. I mean, you got it right in those woods right there at your back door.
Yeah, where we haven't seen the bobcats in a while. We have seen the fox once. But,
well, I hope the cats are doing okay. I kind of like seeing them on the camera. Those are pretty
cool.
Once
in a while we have a cat wander on through.
Yeah.
Really
man,
strange.
Look at the chat, dude. It is hopping, Steve.
Certainly. So let's get to our roll call.
Wow.
And then we'll get to our illustrious guests tonight with us. Of course, We have Professor Kevin Harder, who's down there. Now, good evening, Kevin. And we have Dr. Haskell Hart with us as well. Hello, Dr. Hart. And yeah.
There he is.
So,
Oh, yeah, we got you. Yeah.
So anyway, let's do our roll call in chat. First
yeah.
in today was who stopped in to say have a great show because he's from over the pond and it is 2 o'clock in the morning where he is, I believe.
Yeah.
And so our first two in were from the UK, Relic Hominin. Good to see you. And Sharon, who also it's got to be at least 2 o'clock in the morning where she is in Belgium. So hello, guys. We'll catch you guys on the.
Yeah, thank you for coming in. We'll catch you on the replay. Good to see you in the chat. Thank you.
And we got first in that's still here is Walter Kroll. Hello, Walter. Michael Marcinko, good to see you, sir. Michael? Larry Sharp, welcome again, Larry. Tennessee Cherokee. Bristol, good to see you. Sandra Piper. Grasshopper is in the house. Eyes in the Woods. Hello, Eyes. Good to see you.
Yeah.
Lance Windsor. Hello, Lance. Tennessee Cherokee, if we didn't say that already. Daniel Weeks.
Daniel?
Little Kilroy was not fooled.
Little Kilroy.
Jeff D. Smith. Hello, Jeff D. Good to see you. Boris Kahn. Hello. John, Sasquatch Wizard Adirondacks. Good to see you.
John?
Angel Nolan. Hello, Angel.
Angel?
Brian Wolf.
Brian.
And Brian, good old Brian. Behold, it is he who brings knowledge to the masses. Nikki, Sasquatch Secrets. Hope you're feeling better, kiddo.
Nikki.
I know she's a little under the weather, so wishes to you. Yeah. Blue Ridge Bigfoot. Good to see you, Blue Ridge.
Blue Ridge.
Helton, a.k.a. Faster Man. Chris, good to see you tonight. It was great talking to you last week. Brian and Chewy go hiking. Hello, Brian. Larry Sharp. I think we already called him out. Jay Fritz is in the house. Jay, what's happening? Wild Humans.
Wild Humans.
Squatch talk to me in my head. Yep.
Welcome.
Don Come Out Jr. Good to see you, Sasquatch Research Maine. DM Zabo. Hello, DM Zabo. Good to see you. Jen, the bouncer. She is in the house.
Jen.
Hello, Jen. And Finding the Trackway. Bill, what's going on in the house? I'm sure Mrs. Trackway is lurking somewhere as well.
Yeah.
Buttworm. Sorry I'm late, but you're here.
David we're proud Parker. To have you.
David Parker, Blake Burrows. Hello, David, guys. Brian McMahon. Hello. So we got quite the crowds in the house tonight. I'm still scrolling I'm
scrolling, but I got lost a while ago.
Okay, I think we got everybody that's said something.
Yeah, if we missed anybody, it ain't because we don't love you, because we sure do. And hello to all you lurkers out there. You know, if you want to lurk, that's okay. I do it too. But come in and say hi every now and then. Just pop a pop thing in the chat there.
So I hope the crew has got their thinking caps on tonight, because tonight is going to be a deep-seated conversation. And this is how it all started. Was that last week the Coast to Coast YouTube channel put out a brief half-hour interview with between George Knapp and Dr. Melba Ketchum, and the title of it was Bigfoot DNA, The Shocking Truth. You can see it right up there on your screen. And it turned my stomach because, there's nothing more that is more against our mission here is junk science. And this is not, so this short video I was going to do, just as I was putting it together, I found myself having to explain myself and explain my reasons why. And before you know it, what was 5 reasons why I do not believe the Ketchum DNA study, why I believe the Ketchum study is flawed, seriously flawed. It turned out to be like 30 different points on the 5 different topics. So it just spiraled. So I figured, what the heck? Let's get Dr. Hart back on. Let's get Professor Harder back on. Let's talk about this because a lot of information I've gleaned over the years and some of my um, ideas. I mean, I always had that suspect in it before I met these fine gentlemen. I always felt that there was something wrong with the project, starting with the first couple of reasons why that I bring up in this presentation I'm doing. And gentlemen, if you need to jump in at any time, please do so. Stop me, say, hey, this is wrong, this is not right, or it's something you want to add, please add to it. Um,
Will do.
so first is so we ready with the thinking caps. And by the way, I do have a hat for the Ketchum supporters tonight. No, we're gonna take the tinfoil hat off.
Yeah, I think my tinfoil hat. I think I took it apart because I cooked a baked potato in it.
Well, I wrapped the leftovers in it.
Have to get some fresh foil.
But so again, this is nothing at all on this is personal. This is the reasons why I do not believe the Ketchum study holds any water. So first, to start off, there's some definitions. Now, I did release a video a month or two ago about the 7 terms of DNA terms that a Bigfoot researcher should know. And out of all of those, only one, alleles, is in this definition set list. So get ready to expand your knowledge even more, because some of the terms we're going to be talking about tonight is, for example, STR analysis, which stands for short tandem repeat. Analysis is a molecular biology method used to compare specific regions of DNA between individuals. So I'm not even going to get into the whole deeper explanation, but the graphic is there. You can read it there on your screen. We're going to be talking about reference sequences. Reference sequences refer to a comprehensive, curated collection of DNA, RNA, and protein sequences that serve as a standard or reference for a particular species.
And that's definitely one area where the Ketchum study probably erred in
right.
trying to assemble the sequences into something recognizable.
Yes.
Dr. Hart knows better than I.
And that's, I think that's the reason why it's in the definition, because it's one of the many different reasons. And then we got phylotrees, short for phylogenetic trees, are branching diagrams that represent evolutionary relationships amongst various biological species or entities based on similarities and differences in their genetic or physical characteristics. And then, of course, alleles. Alleles are different versions of the same gene that exist at the same specific location or locus on a chromosome. Each individual typically has 2 alleles for each gene, one inherited from each parent. These alleles can be identical or different, and their combinations determine various traits, such as eye color, blood type, and susceptibility to certain diseases. So, and finally, if we haven't thrown enough of this at you already, we have the single nucleotide polymorphism analysis, which is a method used in genetics to study variations at a single nucleotide position in the DNA sequencing among individuals of species. SNPs are the most common type of genetic variation and occur when a single nucleotide in the genome differs between members of a species or between paired chromosomes in an individual. And the big consideration here is, and this is all going to have relevance down the road, is that large sample sizes are often required to detect significant associations with these SNP analysis tests. So
that's always going to be a problem for us.
Right.
There's always going to be a basic problem needing more samples.
Right.
No matter what, we're going to need more samples to better understand it.
Correct. And as we're going to talk, that is one of the major flaws of the Ketchum study, is that they do not have that large um, uh, sample size. So, so let's go into the first reason why I feel the Ketchum study is not a valid study. It's the reason why, and this is totally not science. Well, it's scientific because the process is flawed and the manner in which the article was published. So, first of all, in November, previous to the quote unquote publishing of the study, Ketchum's own press secretary issued a press release hinting at the findings of the study, and this goes against completely against the scientific process. Science does not come out with a press release stating what they found. They come out with the study, and then subsequent press releases will be afterwards.
Right. Yeah.
So that is issue number one.
Usually they'll come out, they'll publish the article and say, this is what I found, and this is how you can find the exact same thing that I found.
Right.
Yeah.
The second, again, more reason one having to do with the manner in which the article was published was that it was published in a journal owned by Ketchum herself. Although she did try to disguise that at first by just coming out with the blanket statement, we're published, yada, yada. And the mainstream media caught up with her real quick saying, time out. This journal has only had one article and this is it. And wait a minute, who owns it? It's Melba Ketchum. So she got caught.
And there hasn't been too many other articles published in that journal since then. I think there's been like one letter or something like that.
Correct.
And it hasn't gone on to function as an actual journal.
Right. It's not like this was Nature or something like that.
Yeah. But so that was the confusing thing for me was that because I listened back to the interviews, and she says that it made sense for them to purchase the journal so that they could retain the passing peer reviews. And
yeah,
I don't understand that. I'm not sure how that works really.
Well, that's because of reason number two was the study failed peer review.
Yeah, they were not. I mean, I guess she was trying to say that one of them was lukewarm. Like we could maybe publish part of this if you do these things.
But the funny thing is that one was never published. In other words, she put out the rejection letter. But she never put out that one where it was that was only her word saying that, basically. And that makes me quite dubious. I mean, already we're at the level of dubiousness as we stand by the way this has been released.
I guess the ones that she was referring to as passing were the ones from the James, it's abbreviated JAMEZ, the Journal for Advanced Multidisciplinary, something, Advanced Multidisciplinary something research.
Right.
Anyway, yeah. But then they had to rename it after they. Yeah. It doesn't make that was a confusing thing for me. Is like you had to purchase the journal to preserve the peer reviews, but then you're renaming it, and so it's like starting out as a brand new journal. So why did you?
Right.
Yeah.
Well, if I could jump in here. Yes, that particular journal that she claims passed peer review. Really, they required her to do some things that she didn't do, as I think Kevin pointed out. One of the chapters in my book deals with the peer reviews. And actually, before I even had seen them, I came up with about 10 issues or more. And believe it or not, every single one was also brought up by at least one or more of the peer reviewers.
Right.
So this was an independent. It really made me feel good that I was in with the same thoughts, and there's some serious objections there that anybody who's really into this needs to dig into, because it's not. She tried to dismiss it all as what, hate or
Oh yeah,
what is the word she used? She had another term for it.
conspiracy.
Yeah, conspiracy and all this.
Yeah, the Galileo effect.
That's
Yeah.
nonsense.
Yeah, yep. And the funny thing is that, and again, here's a shameless plug for my new book. I talk a lot about illusionary superiority in my newest book, The Psychology of Bigfoot. And at the very end of this, we talk about some of the reasons for her rebuttals. And we'll get into all the psychology of that particular thing about her because of hate and because of conspiracy and all that utter nonsense. And we'll break that down a little bit better in a bit. But
Nobody hates on Dr. Ketchum. No, it's nothing personal whatsoever.
this is just the facts.
Right.
Just the facts. Next slide. So, reason number 3 was the study. That did not advance on that. Whoops, sorry. I'm advancing it on the wrong page here because my advancer is only supposed to advance on one item, but for some reason, if I click on this screen, it starts advancing on another screen. So I got to constantly move this.
Now, for everybody in the chat, I've got my screen maximum size, so I can't see the chat. So just make sure you type your questions in caps, and I'll go back to it. Right now, I've got it blown up where I can see without reading glasses. So that's very big.
That's what I'm doing.
Reason number 3, the study's process was flawed. Lack of peer review and publication in a reputable journal. The paper was not subjected to the rigorous peer review, which is a cornerstone of scientific validation. It was published in the journal De Novo, which appears to have been created specifically for this paper and lacks the established reputation of mainstream scientific journals. The absence of peer review raises concerns about the quality and objectivity of their research. So any peer review they did happen, it basically failed it, or there was expectations in order for us to pass this, you need to do this, and it was not done. So that's another reason why the whole process is flawed on this study. And this has a common theme. And as we get towards the end of this presentation, which we should be done with the presentation, not too bad of shape. We're going to have plenty of time for questions and discussion. So, the end game here, you'll understand how this all ties in together.
And as Dr. Hart already pointed out, there was a chorus of people objecting on similar grounds, for similar reasons. Right. So it wasn't just like one person that didn't like her.
And it's a lot of times, and I don't, and I don't know how scientists do their processes because I'm an investigator. But a lot of times I will not look at investigation notes of another investigator until I do my own investigation to see if there's any parallels in the investigations. And then when I compare the notes, I go, aha. And that may either lead you to you're on the right track, or one or myself or the other investigator is on the right track or the wrong track. So
that's important. I think for most journals, probably the peer reviewers are independent, and only if there's serious issues does the editor get them together and talk about it. And I tabulated all the objections as I specifically as I could, and it's interesting to see that numerous peer reviewers had the same thing to say on every point,
Right.
just about, including me, which I was Johnny come late. And so, that says a lot. These people have I don't have any reputation in genetics. These people were presumably, I could tell from the comments, well qualified and knowledgeable about the subject.
They're
I kind of wish,
DNA nerds.
yeah, I kind of wish they had gone a little farther and done some
Right.
of the searches and things that I did later, and it would have put her to rest for good. But nevertheless, they had really good comments, I thought.
Now, the other thing is the sample collection and contamination concerns. Samples used in the study were collected by various individuals and groups, raising concerns about the chain of custody and the potential for contamination. And part of that, I think, is because there was no descriptors or no chain of custody necessarily involved with the study, which you would expect. Um, I know in a criminal case, when there is evidence, especially of a DNA background, there has to be a very clear cut chain of custody. And one of the biggest problems with the O.J. Simpson case, if we go back to that study, I mean, I was one of the few people. I had a job where I was monitoring stores, and we had radios in all of our offices, and all of them were tuned to the trial. So I would go from one store to the other and pick it up. And meanwhile, my boss was in his office listening to the trial. And when something would happen, my phone would ring, and my boss would be like, "Hey, did you just hear this on the trial?" So we were constantly calling each other, and amongst all of us, we were all talking about the trial. But one of the major concerns was the chain of custody of evidence, particularly with a vial of blood that Detective Phil Van Adder had placed in his shirt pocket. And instead of sealing that up and sending it off to the lab, he had kept it in his pocket. And he proceeded to go to the Simpson household, OJ's actual household on Rockingham. And because of that, it let open the door for a potential contamination of one crime scene to the residence of the defendant. So that's why the documentation and chain of custody has to be maintained in studies. That's why Dr. Orcutt, when you send him a sample, he will send you instructions on how to package it, label it, and that way he can maintain that level of that chain of custody of evidence. So that way, when the report comes out on a particular sample, if it's something remarkable, that chain of custody will be present in the report. This was not done with the Ketchum study. Therefore, it leads to questions and concerns about contamination of the evidence, especially given the results she would come out with.
Yeah, just looking at the large number of samples, 111, I think, and the many different people who collected them, I'm very certain that they didn't use forensic techniques that Steve, you would require. I mean, they probably, when they collected the samples, they didn't even know that they would be submitting it. They submitted it years later in some cases, and other questions.
And the whole study really wasn't planned in advance in the way that the Orcutt study is being planned now, or has been planned. Instead, what became known as the Ketchum study kind of grew organically as people found out through word of mouth and wanted to had something in the freezer that they always wondered what the hell it was,
or sitting on a shelf or in a jar somewhere, and so already that.
So there was guaranteed to be problems of that sort, I think, in this, because it wasn't planned in advance. Really, it was an organic kind of expansion of. She started out testing a few samples for some individuals, and it grew. And
I Ganir, I wholeheartedly agree with you. We should not mention her name or her work anymore when proof is talked about. 1000% agreed.
Well, the sample collection was kind of weird too because I checked into that, and I think there was some. It was like she was lawyered up. If you submitted a sample to her, you had to sign over all ownership and rights to the sample or whatever, and whatever she found belonged to her.
Right.
It's like a money grab right from the beginning. If you've got Bigfoot DNA, it belongs to me.
Oh, and by the way, it's going to cost you $2,500 to have your sample included in the study.
Yeah, well, no, that's
And-
Now, just speaking about the hair samples, of which there were most of the 111, it is possible to decontaminate the surface before you break it up and analyze the DNA inside in the medulla. And this is reported and done very successfully by a place called Mito Technologies. It's up in Steve's neighborhood,
Yep.
New York there. And, she said she washed them and vortexed them in alcohol water. Now, I don't know the proportions, but DNA is not soluble in alcohol. It is in water. And it's possible she didn't get it all off the outside of the samples, therefore. And so, along with the fact that sending them to laboratories that deal in human DNA with human primers is only going to give you human results, if any. And, you can have a hair that's from a bear and human contamination on the outside. And if you don't clean that up, the lab's going to give you a human result.
Right. Yep.
It seems unlikely that out of 100 and some samples randomly collected in the woods, that all of them would turn up to be human.
Now,
It's just not possible.
right.
You
Yeah,
know,
exactly.
these are remote places. There's lots of other animals around and not as many people. So that tells, well, it's not a proof, but it's
there's a
indicator.
definite problem with those odds.
Yeah,
Right.
that's right.
Well,
And
very, very similar when I was talking about the Homo naledi, how they were saying that none of their bones showed any type of traumatic injury. And I found that very odd because they had all different ages in that cave. And it sounds like to me that those subjects had died because of some kind of external event, like being trapped in the cave and end up dying of starvation, or maybe
drowned, or they could
a
have
gas
been-
release that killed them, and that's why there was no more. Because one would think that if you have 15 different bodies of all ages, there would have been some type of, at least one of them would have been predated on, or had some kind of wounding to them. So that's very similar to this. Is how come? And I said it since the very beginning that I believe that it was the primer. She was using a skewed primer, which caused those results to be all the same. So Kaiju Ninja asks, where did Ketchum get the samples? Was it scat hair? I know a lot was hair. There was a toenail. There was some.
The samples were submitted by many different people, individuals and groups from all over the country and Canada as well. So it really wasn't a standardized process.
Now, I don't want to necessarily get in the Justin Smeha particular sample until towards the end, but that can be a side discussion because there's a really interesting story behind that. That Smeha actually had Dr. Brian Sykes analyze that sample as well.
For his boots,
And-
yeah.
And
I'm sorry.
the background was, and this is out of and let me just go to that sidebar conversation now, was that when the study was in process, I was very close with a few of the people that were involved with the study. J.C. Johnson was involved. I know Autumn Williams had her ear close to the ground, and I was talking with both of them. So I knew a year and a half before the results came out what the results were alluding to or going to allude to. And I knew, and I was preparing myself for these issues since day one because it did not sound correct. But one of the whole Smeha sample was that Smeha put that sample in the Ketchum study. He was getting tired of waiting. So he had Brian Sykes do the DNA and it came out as bear. But everybody within the study group had been told by Dr. Ketchum that it was, this novel human, this novel hominid, or, this hybrid. And the fact was it was bear. And
Yeah.
that's why Smeha was calling her a fraud and a charlatan before the study came out because he knew what she was telling him, but he had a real deal scientist tell him, no, it was bear.
Yeah, and of course she responded saying that he submitted a different sample
Of course.
than the one he sent to her. Also, two other forensic wildlife labs analyzed supposedly a cut from that same sample, and they came up with bear as well. One was sponsored by Bart Catino, and one of them sponsored by Taylor Huggins.
Right. Yep.
And
The
they
one I
used,
remember that was a very large sample. They called it the steak.
Yeah.
yes,
Yeah.
yeah, it was flesh as well
Mhm.
as hair and skin. Yeah,
That's
And-
another. That's
So
another
you-
confusing factor that we should maybe mention right now, because the nuclear sequences were not recovered from any of the hair samples. Right, it was only
No.
it was the steak. The steak
Yes.
supplied one of the nuclear sequences.
yeah, from the meat of it. Yeah,
Yes, and so the very best samples as far as the information that they could contain, were the hair samples. But unfortunately, they were not able to recover any nuclear material from the hair samples. So that so that's an added complication. The samples that supplied the nuclear material were of different kinds. What were the other ones?
here's where we get back to Steve's comment about chain of custody is very important. When that sample was collected, all this should have been documented as to who cut what off of it and gave it to whom, with signed statements, pictures, and so we're sure it's all from the same sample. That's she's got a possible case here. I mean, I don't think they can prove that this was all from the same sample. They say it is, but I guess I trust Bart and Tyler more than I trust Melba on that. But
Yeah, correct. Yeah, I would.
Yeah. Sasquatch Wizard Antibiotics. The one blood sample was reported to come from a Sasquatch punching through an irrigation pipe to grab a skunk.
yeah, yes,
That was J.C.
that's-
Johnson's. Yeah.
that is sample 140, which turned out to be a dog. I mean, apes don't bite on metal pipes very much. Dogs bite everything,
Yeah.
So that's not proof either, but I totally analyzed that, and it's a dog. I've got figures to show it in my book, colored pictures.
Speaking
all
of
the
which,
different- yeah.
Steve, lizard
Professor,
man case.
that's a cat, Professor.
Yes, my cat came to visit. Sorry. What was the other? Yeah, so that was the blood sample and the steak sample, and the third nuclear sequence came.
It was some saliva off of a food trap in Alabama, and that one came out. It was contaminated. She said there was no contaminant. The highest, greatest hit with the largest number of bases was a fungus. And of course, that's, neither here nor there, but it was contaminated by a fungus. And another one, a bacteria, but then there was a lot of good human DNA. And she found it matched an African haplotype. And so who knows? I mean, all these hair samples, you have to consider them like an environmental sample because you don't know what touched it. For example, a sloth is said to have DNA on its hair from 800 different species. A lot of them are microbes, of course, but I mean, any piece of hair out in the environment is almost by definition contaminated. The animal touched other animals. Other animals may have walked across the hair or spit it out or whatever. You don't know.
Or the animal may have traveled through an area that
Sure.
had other animal DNA on it.
Right. Yeah. You have to assume that to start with.
And that's one of the inconclusive nuclear DNA findings. Paper reports novel nuclear DNA sequences in the Sasquatch samples, but the interpretation of these findings is problematic. The authors of the Ketchum Study suggest that the nuclear DNA is a mosaic of human and non-human primate DNA, but this conclusion is based on limited data and lacks robust statistical support. The possibility of contamination or sequencing errors cannot be ruled out.
Well, I do believe that she took the results of a search against the database, and because the genes they selected are conserved and aren't changed too much from one species to another, they got a hit list with a lot of different animals, and therefore they said it was a mosaic of those. That is not the way you look for hybrids. You've got to look at the sequence and look for regions that are from one parent or the other. You can't take a hit list and say that everything on that is part of a hybrid.
Right.
That's not the way you do it.
Right. Next slide. So, and then again, lack of control group. The study did not include a proper control group of known primate species, making it difficult to compare the DNA findings to established genetic data. The absence of a control group limits the ability to assess the significance of the novel sequences observed in the alleged Sasquatch samples. And the reason why is confirmation, to me, is confirmation bias, because she at the very outset wanted to believe that this was a people or a homo type of background, that is somehow they were a people, and that was her standing before the study even came out. And I think that, to me, it seems a lot of this study is I am going to fit my evidence around my theory instead of fitting the theory around the evidence.
I think you're absolutely right. I think I called it somewhere in one of my other interviews a preconceived notion.
Yeah. And that's, unfortunately, we'll see towards the end of this how illusionary superiority works hand in hand with confirmation bias. So, and then there was overinterpretation of data. The author appears to overinterpret their data, drawing conclusions that go beyond what the evidence supports. The claim that Sasquatch is a human hybrid is based on limited genetic data and lacks a clear evolutionary explanation. The possibility of alternative explanations such as contaminations or sequencing errors is not adequately addressed in the study. And again, reason, confirmation bias, is that I'm going to not look at this because it goes against what I believe. And we see this as a common thread in this study.
The other sort of subtext here, maybe. That's not really the right word, but anyway, the other issue, kind of underneath this, is the question of uh, how species are related to begin with, and I think she's coming at this from the assumption that um, species are created separately, actually, and or in other words, as she has admitted, the whole human hybrid idea is really contrary to evolutionary theory, and I think I think that's why she liked it. I think that's why she promoted it so readily, is because she herself is hostile to the whole evolutionary idea.
And again, another reason, lack of transparency in data sharing. Paper does not provide full access to the raw data and analysis methods, limiting the ability of other scientists to independently verify the findings. I know, Dr. Hart, you managed to end up getting your hands on some more of the raw data, and the lack of transparency raises concerns about the reproducibility and validity of the research.
Yes, she did publish the nuclear sequences online, and she has a website, Sasquatch Genome Project. You're probably all aware of it. And so that's what I got, where I got that. And her paper in the supplementary tables and stuff do list the mito sequences or the mutations thereof. And so you can check, but the thing is, she never published any of her search results,
Right.
to prove her points. And of course, I went into this in detail, and it's kind of suspicious that she wouldn't do that, because you have no idea how she came up with these conclusions.
Well, I think one of the final points I bring out in this presentation by an independent group, and we'll talk more about that in a bit, kind of alludes to why that never came out. Because what I'm seeing here, and it's kind of funny because I'm a psychology guy, I'm a behavior guy. So I'm looking at this study, reading the issues with that, and I'm beginning to look at this report we're going to be looking at the end of this presentation, going, I'm beginning to see correlations here. I'm beginning to see how this was bad from the start, and I understand the whole thing now. I understand the person's behavioral patterns now. And so it's not a surprise to me why that's not there. And I'll explain that towards the end. But reason number 4, misinterpretation of results, inappropriate use of reference sequence. The use of human reference sequence for nuclear DNA analysis biased the results towards conserved human genes and limited the ability to identify potential novel sequences. Inconsistent and incomplete STR analysis. There's one of those definitions. The short tandem repeat analysis showed numerous anomalies, including failed sequencing, unknown alleles, and inconsistencies between different tables in the study. The study also failed to recognize dog DNA in sample S26. Bizarre phylotrees. The phylotrees presented in the study for samples S31 and S140 included distantly related species like fish and chickens, suggesting errors in the analysis or interpretation.
And the fact they never even looked at their own figures. I mean, good grief, we're talking about a primate here. And I think she just turned it all over to this guy in, where is it, North Texas? And he just ran the computer thing, and that's what he got. And nobody thought to look at it. It's unbelievable.
I mean,
Harp and chickens and all that stuff. I mean, that doesn't belong in a paper like this.
Well, my whole thought of that too is remember who she's selling this to. By this time, she has got herself a gaggle of followers on social media.
Oh, yeah.
Right. Which, again, we're going back to illusionary superiority. It kind of boosts the ego. And they're not educated as to the science of DNA. So she can put whatever she wants they're not going to read that with a fine-tooth comb, right? They're going to take her word for it. So
Just
it
like
doesn't.
you have to take the word of the 23andMe people when you send off your. I mean, you send it off and they tell you what it is.
Yeah.
I mean, the good news is that I had several years of experience of this before the Ketchum study, looking at DNA reports that were skewed. I had one particular Bigfoot group say that this skeleton they had found was this novel thing, and that it had never been. And what it turned out to be, if you read the fine print, it was Homo erectus, and they were trying to pawn it off as something else. But all you got to do is read the fine print.
Well,
And
and yes, getting back to 23andMe or whatever family tree DNA, I think she used, if you pay for it a little bit more, you can get your whole mitochondrial sequence, which I did. And actually, I ran it through programs and I came up with the same haplogroup that they did and the same 4 extra mutations. So not everybody pays that little extra for that, but you can get your full sequence. And if you don't like what they said about you, your heritage, you can check it out.
yep. John brings out a point. Steve in the panel, it would appear Dr. Ketchum became emotionally attached to the results. But can we talk about the rest of the individuals' hard work? Well, actually, yeah, absolutely. Listen, the bottom line is this: Professor Harder said earlier, is that a lot of these samples, these people had already collected them, and they had been sitting on a shelf, or they had been in storage, or they've been in somebody's freezer for years. And now the study came out, and they grabbed it and went out. It has nothing to do with the integrity of the individuals that collected the studies. This has nothing to do with the people that collected. They were hardworking, they meant the best, and they collected what potentially could be DNA evidence. Unfortunately, it seems like they struck out, but that happens. I've struck out. It happens. It's all we need is one sample. It's going to be hard to find, but the bottom line is this: is that all the individuals involved all had good intentions. The only thing that got twisted was the person in charge of the study. That's where it got twisted. So this has no bearing on the people who collected the samples. Mind you, these people collected the samples 2007, 2008, 2009, 2010. These samples were collected a long time ago, before we knew as much as we know about DNA today. We weren't talking about DNA collection methods in 2008 or 2010 for that matter. We didn't start talking about that until 2012, 2013. So these people collected the samples doing the best with what they had and what they could do. And now you can go on eBay or Amazon and buy yourself DNA collection kits. You can buy yourself buccal swabs. You can buy yourself tubes to put the buccal swabs in. You can buy yourself latex gloves. Some of those things weren't readily available back in 2008, 2009, 2010 when these samples were actually being collected. So the best we generally had were gloves.
DNA science has really come a long way in a short period of time, too. Before we didn't have, back when I was in school in the 90s, we didn't have like supercomputers that could process a DNA sample and break it down. All this stuff is really cool.
And mind you, we are applying 2024 standards to this study. That the study came out in 2013, beginning of 2013. So we are applying standards that are 9 years now down the road. But even then, scientists had problems with contamination, obviously because of the results. So then we have contamination concerns again. The study denies any contamination in its samples despite evidence of extra mutations and mixed haplogroups, suggesting the presence of multiple DNA sources. The decontamination procedures for various sample types like blood and tissue are also unclear. Misinterpretation of the HVR1 results. The study claimed all 11 samples showed 100% human mtDNA, but the HVR1 analysis indicates only 3 out of 11 samples could be definitively considered modern human. Misinterpretation of MC1R results. Study attempted to correlate red hair with the specific mutations, but found these mutations in a black and white hair sample, highlighting a potential misunderstanding of the genetics of hair color. So,
Big one.
yeah, that's a huge one.
Yeah.
And hair color is really not a key indicator of anything because
Right.
orangutans have red hair, people have red hair, Sasquatch is said to have reddish hair in some cases, and so that's kind of a sideline. It doesn't prove anything.
Well, since all her samples end up to have human DNA in there, I'm wondering if it didn't have something to do with the primer she was using, or super
absolutely.
secret. Right.
Well, yes, because they sent it to a lab that does this DNA testing for the populace, and they only use human primers because only humans send them samples.
Right.
And what I did just to check the system, I submitted a horse, a cat, and a dog sample from friends of mine. I don't have pets, but and I just pretended it was regular, use their kit and send it in like a human sample. And all of them failed to amplify, which proves that, their human primers are not going to find another animal. Okay? There was plenty of DNA. We took this horse and really swished it around, and it failed to amplify because the primers were human and not general enough for these other animals. So that was interesting. I'm sure they'd not be appreciative of me sending in animal samples to their human lab, but since they're using human primers, they won't show up anyway.
Well, at least they didn't come back as cat man or dog man.
Exactly.
Well, that's right. When I had a conversation with Melba when we were still talking, I said, "Have you ever seen dog or bear DNA in any of your samples?" And she laughed and said, "Well, maybe it's dog man." That was her answer. I'm not kidding you.
And she's still in that mindset, as if you.
I mean, she tried to brush it off. Obviously, to me, she has seen it.
Right. That was a non-answer, right?
That's right.
Circumvented it, and that from an interrogator standpoint, I'd be like, well, that's a yes.
Oh gosh, she blew it right there. I mean, that tells me that yes, she has seen it, and no, she doesn't want anybody to know that.
And that is probably the most aggravating point of it. We expect her to act like a scientist, and she's not. She's acting like, and we'll get more into that too. What some of the examples of who she's acting like. Again, more stuff from Dr. Hart's study, misidentification of samples. Study misidentified samples S26 and S140 as hominins, while independent analysis and the author's own assessment indicate they are a black bear and dog, respectively. And that's a huge one. Lack of control samples in SNP analysis. The single nucleotide polymorphism analysis lack control samples from known non-humans, making it difficult to interpret the significance of low percentage matches and distinguish between degradation in non-human DNA. And then, of course, false conclusions about novel DNA. The study's claim of novel DNA structures based on the presence of single-stranded DNA is refuted, as single-stranded DNA can result from degradation is not indicative of a new species.
Right. So, not only that, it probably would not survive. it wouldn't be able to replicate, for example. I mean, no known species has ever come up with single strands. Viruses are the only things that have single strand, some of them, right? Check me out there, Kevin.
I think that's right. I think that's right. And they're not even alive. Technically speaking.
Yeah. I mean, I guess this is Occam's razor, the most likely, simplest explanation is usually the true one,
So we just have an interesting comment from Dirty Woods saying, how does Bigfoot get enough calories without being able to cook meat? Well, number one, you don't get calories from cooking something.
Yeah.
Actually, there's in a lot of cases you lose calories when you cook something. That's number one, because you're burning the fat off the meat.
Yeah.
So you actually lose calories when you cook something. But there are stuff like deer and deer liver, which is huge amounts. So how does Bigfoot get enough calories without being able to cook meat? Well, how does a black bear, which needs 15,000 calories a day to intake before hibernation, how does that take that much in when it doesn't cook its meat either? So let me ask you that question back at you.
Or a moose, which is huge and it's a vegetarian.
Right.
Yeah.
And then how can they collect food all day and not get caught in a trail cam? Are you that naive to think that there are trail cams everywhere?
Well,
how many times have I put trail cameras up and caught zero? Nothing. I've been in my research areas. I've planted cameras that have been out there for weeks. I don't even get a deer on them. Why? Because when you put a trail camera out there, if say you put 5 trail cameras, that's 5 needles in a big haystack. And in my case, it's a 2.5 million acre haystack. So that answers those questions. Let's move on to our discussion. So reason number 5, and this is stuff maybe you guys don't necessarily know about, but she was found to intentionally ignore standards by the Texas Forensic Science Commission. So what they did was they investigated a complaint against Dr. Velva Ketchum for providing expert testimony on canine DNA analysis in a 2010 capital murder case. Now, mind you, 2010, this is the same time the study's going on as well. So here she is, finding the thing that I'm so important because I testify in court as to DNA of animals, and I'm in a murder trial and all this. The commission found that Dr. Ketchum was professionally negligent in failing to obtain accreditation for a laboratory before performing forensic analysis and offering related testimony. Again, illusionary superiority. She feels he's above that. That's my feeling on this. The commission also found that Dr. Ketchum's testimony in the Schmidt trial was incomplete and misleading, because she had failed to provide quantitative statement about the significance of her findings. Doesn't that sound familiar when we're talking about that whole sampling? The commission concluded that Dr. Ketchum's testimony constituted professional misconduct because she was aware of and consciously disregarded an accepted standard of practice. And again, we see that throughout the Bigfoot DNA study. If she is not willing to use accepted standards of practice in a capital murder case, do you think for 1 second she's going to apply any standards to a Bigfoot DNA case? Absolutely not.
Well, that's showing a definite pattern.
Sorry, go ahead.
I said that was showing a definite pattern. She's not willing to play by the rules, number one, and she thinks she knows better.
Correct.
Rather than what's reality, she's going to create what she believes.
Correct.
Yeah,
yes,
the matter of the criminal case, I'm thinking it maybe involved STRs or something like that. And when you say a person's DNA matches somebody else's, you have to say, how likely is that to happen on a random basis. Okay. I mean, just because it matches, there are other people that match as well.
And I believe, Dr. Hart, that she was
Am
matching.
I right there, Steve? Or you?
Yes. I think what the particular item involved in this particular case was that they found some dog hair on the crime scene, and that dog hair was sent to Melba Ketchum along with a sample of the defendant's dog.
Oh,
and
yeah.
she stated that this dog hair matched the DNA of the dog hair there with the defendant's dog hair, and that's how this all came about. That was her testimony, and because there was some lacking of accreditation and disregarding standards of practice and failing to provide a quantitative statement about the significance of the findings, all of this constituted this professional misconduct and being professionally negligent.
There are probably maybe hundreds or thousands of dogs that might match that DNA. Yep. And you have to say how likely is it that
correct
that it's the one that you're looking for versus a random.
And it's either one of two things. You're seeing somebody that's I think swept up by the prestige of being able to testify in a capital murder case for the Texas Forensic Science Commission. And I think that number two is you have somebody that feels above principles, or above standards of practice, like she knows better. So I really think that she was out of water. I think this was above her head. All of this DNA stuff is above her head. Unless you're doing some simple DNA, that she was a cat vet. She was doing cat DNA.
Yeah, she was definitely out of her depth in trying to identify an unknown species.
Yeah.
And obviously she was out of her depth going into a capital murder case.
Yeah, Kevin, you hit it right on the head. I think what she did before, and I've read a little bit about it, is, somebody's dog bites a horse. They collect the saliva. Whose dog is it? Kind of thing, It's a much more controlled thing than identifying something that's unknown.
Yeah. Right.
Yeah. And the same with all her laboratories and all the people she worked with. They were not experienced in this kind of work.
Right. And they expected it to be a simpler, more definitive kind of process.
Yeah, I don't think they realize how difficult it is. And I've heard some of these experts on this, people who've, for example, sequenced Neanderthal DNA and stuff, and are really the cream of the crop, and say how difficult it is to avoid contamination and other issues like this that I think she just overlooked.
So here's the few other odds and ends, which is based on Professor Harder's work, was the designation of Sasquatch as a human hybrid. If the data were correct, the label human hybrid overstates the human component in the Sasquatch's genetic makeup. The term hybrid is misleading, and that Sasquatch should be recognized as a distinct hominin species. That's if her study it was in the correct column.
Yeah, there's no. It's a confusing point, but honestly, there's just no good reason to refer to it as a hybrid, even if it does have human mitochondrial DNA. That's just not what a hybrid means. And honestly, but it is confusing because, if the mitochondrial data are accurate, and if they really did come from hair samples which were not human, the most you could say about it is that they are descended from hybrids, but there's a huge difference between being descended from a hybrid and being a hybrid species, right? Being descended from a hybrid just means that there's this one small detectable change, and it's only detectable at the molecular level, or that's there's no reason to expect it would be detectable otherwise. I mean, it's a very tiny, the human component that she's talking about is just vastly overstated, I'm afraid.
Right. Again, I think it has a lot to do with their own confirmation bias.
Yeah.
Conflict with evolutionary theory. The Ketchum Study's conclusions are at odds with the established principles of evolution, particularly the concept of common descent. Neglect of the unknown progenitor species. The Ketchum Study proposes that Sasquatch is a hybrid of humans and an unknown hominin species. Study fails to adequately address the nature and identity of this unknown species, which is crucial to understanding the origins of Sasquatch. The recent origin of Sasquatch. The Ketchum Study proposes that Sasquatch originated relatively recently, around 15,000 years ago. This time frame is implausible, and that Sasquatch likely has a much older evolutionary history. We talked about that a few weeks ago with when we had the Quest for Fire episode of Squatch-D TV, where we talked about Homo floresiensis and Homo naledi and all of that. And we started in like Homo habilis, which Homo habilis and Homo naledi show inline toes with some actuality of midfoot flexion still, even though they had arches. So that indicated, and that goes back a couple 100,000 years. So that means that Sasquatch probably would have broken off before that because, or around that time because of that midfoot flexibility and the inline toes.
I'm sorry, go ahead.
But Homo naledi had already started to develop an arch, and their foot did, the pivot point of the foot was more to the front like a human foot, whereas Homo habilis, very similar. They were starting to develop the arch. They had midfoot flexibility, but inline toes, and they too were developing. So my guess would be that Sasquatch probably brachiated somewhere before that 235,000 year mark, or at least the an ancestor of what Sasquatch would be.
And the 15,000 year figure is based on one of the more numerous haplogroups, one of the more numerous mitochondrial haplogroups, and there's just no reason to call that a new thing. It's based only on the mitochondrial DNA. I think that's a basic misunderstanding throughout the study because, scientists do classify species with reference to mitochondrial DNA, but they don't. They base that on a known specimen to start with, right? You have your known specimen to start with, and you find out what the mitochondrial DNA is, and then identify it because there was never any question about the identity of the species to begin with. But we don't have all of that for the Sasquatch, and we don't have that. So there was no. I'm still at a loss to explain why she seizes on that so confidently as the beginning of a new species, because there's just no justification for saying that. Even if the mitochondrial data is accurate, there's just no good reason to say that.
So my final portion of this is the behavioral aspect of how Dr. Ketchum has responded to all of this, how she acts publicly. And to me, it's illusionary superiority. She, of course, that's the reason why. The reason why she calls it a hybrid and sticks by her story, because I am Dr. Ketchum. I know the best. I did the study. So it's an illusion of superiority over everybody else. She downplays his criticism, which she does. Minimize people who have illusionary superiority, downplay criticism. They may minimize the validity of criticism, viewing it as unimportant or irrelevant, discrediting the source. They might question the competence or motives of the person providing the feedback, dismissing it as biased or uninformed. Counter criticism. They may respond by criticizing the person who gave the feedback, turning the focus away from themselves, surrounding themselves with yes men. They may gravitate towards people who reinforce their self-view and avoid those who challenge them. And we've seen that. Sally Ramey, who was originally the press secretary for Dr. Ketchum, had a very big dispute over something with her, and she broke off saying, "Hey, this study is something's wrong with it, something's off with it," and she took in this person by the name of Robin Lynn Forest People, again, yes men, as her new propaganda minister. Stagnation, the inability to accept and learn from criticism, can lead to personal or professional stagnation, as they may miss opportunities for growth and improvement. And if you actually read that forensic, that Texas Forensic Commission document, they had advised her that she needed to get certified for this beforehand, and yet she did not. Again, had she taken advice of any of the peer reviews, we might, and the conclusions were the same, we might not be having this conversation. But we are, because she refused to accept any criticism whatsoever. She refused to take any of the suggestions given to her by the peer reviews. And what happened was she missed opportunity for not only personal growth and learn a little bit more about genetics and DNA, but she made the study weaker. She could have strengthened her study if she had followed through with some of the peer review suggestions. And that's the way. And some of the examples we see there, Melba Ketchum writing to the Cron, which is a science-based website, saying one, there is a conspiracy to suppress our findings. Well, there you have it. Here comes this paranoid, there's a conspiracy type of background. This is unbelievable. And then talking to the Huffington Post, she's talking about a peer review done by another scientist. This is unbelievable. My study is a legitimate study up evolutionary superiority. There's no credibility in his study whatsoever. There's jealousy out there. Again, I'm gonna attack or counter criticize the person that's criticizing my work. I'm gonna make it personal and say that person's jealous rather than actually refuting as to why they're wrong. I'm going to instead attack their personality.
Steve, do you have any idea who that person is? Is it me? His study?
No. It was somebody else. It was a science writer, I believe.
Oh, okay. Yeah.
That was
Okay.
writing
I was
about
just
her
curious.
work. And she turned around and snipped at him.
Just jealous.
Right. Yeah, just jealous. Okay. So, again, these are the same statements we hear from people like Biscardi and the Stanlings and Sasquatch Ontario and Cat Hansen and other hoaxers. We hear this from hoaxers all the time because hoaxers are self-imbued with their own illusionary superiority. It's a common trait. I'm not saying she's a hoaxer. I'm saying that people fall into these categories that come out with this false information. And that's part of the reason why is because of this whole thing called illusionary superiority. But anyway, so we can open up the floor for questions, Chris. And gentlemen, if you have any comments, please chime right in.
Somebody had that. Oh, let me get up here.
Well, I have a question. I looked back on Coast to Coast previous podcasts, and I could not find this recent one you say came out within the last couple weeks.
It was on their YouTube channel.
Okay. Well,
It came up on. Yeah,
maybe they haven't republished it yet.
Maybe they took it down because the show has been advertised for about a week.
Yeah,
it's still there, but I couldn't find it at first either. But if you search Bigfoot DNA,
okay, all right, I'll try again.
Well, I think we might have covered some of these, but Kaiju Ninja 1985 says, Steve, where did she get the DNA samples?
They were submitted to, she didn't get the DNA samples. They were given to her by researchers who had collected the items over the years. They included hair, they included, I know, a toenail. There was some blood samples.
There's a huge table of if you dig into the footnotes on the website, there's a huge table there of the submitters and all the information about them.
Sasquatch Seekers. Nikki said, didn't Ketchum say she cleaned all the samples? Yeah.
And that is acceptable, but because you can clean a DNA sample. But again, as Dr. Hart referenced earlier, that we're not sure because DNA is soluble in water, but it's not soluble in alcohol, and we do not know necessarily the processes in which she used to clean the samples, which should have been listed as part of the study, is how she went about and cleaned those.
Well, she said she vortexed them, but actually the way they do it at Technologies is ultrasonication, which I think would be a much preferred method of.
Now, was that process available back in 2010, 2011, 2012?
This paper was written, and I do have a copy of it. It might have predated it, but this is not rocket science, okay? Right. This ultrasonication's been around for decades, so has vortexing. And so whether it was 2010 or later, it wouldn't really matter.
Yeah, I just wanted, not being a DNA expert, I wanted to ask because I wasn't sure if that's
the paper, I'm not sure when it came out. The process of ultrasonication is way older than that.
Don Gumow Jr. says, what do you guys think about the water samples study?
I think he's probably referring to eDNA.
Yeah, I don't know. I'm doing one. I don't know whose study he's referring to, but I've collected samples in the Ouachita National Forest, and I'm in the midst of interpreting them. I'm going to give a presentation at the Texas Bigfoot Conference in October.
Very nice.
And before that, I will issue briefly before my overheads with some comments for people. And I'll make sure you guys get them if you're interested.
Oh, yes, absolutely.
In case you can't be there, which is, in Northeast Texas. But,
yeah, Jefferson, Texas, beautiful little town.
That's right. Yeah, that's right. So
I spoke there back in 2020 at the Texas Bigfoot Conference. Beautiful events, beautiful people down there. You'll have a great time.
And Dr. the samples that you've been gathering, are they water samples?
Yes, from streams and in one case a river. And some ponds in the forest there. I will when I write this up formally, I'll give all the locations. I'd rather not do it until then, but I'll just say so. It's Lafleur County, if anybody's, and it's a well-known place where other groups have gone for expeditions and so on. So.
As the technology advances, I am really excited for the eDNA study. That's going to be so cool.
I am too. And I wondered if the person who asked that question has another study in mind, or which water samples is he referring to?
I don't know. I'm not sure. I think the Olympic project may be doing some of that as well.
Okay.
Not entirely necessarily with water samples, but I do believe they're doing some eDNA work.
Oh, yes. It was connection with those nests. They collected soil. At least that, if not more.
Right.
Well, I think it has a lot of promise. It's got some issues and some limitations, but it's a good avenue for a survey of an area. You collect a dirt sample. It's just that one little place, but a water sample, you get, depending on how big a stream, you could get the whole watershed of things washed in, Yeah. On the other hand, that makes it harder to interpret, too, when you get all that stuff. I found dozens of species, mammals, birds, fish, reptiles, amphibians. And
is that using all mitochondrial markers?
yes, our 12S RNA, 12SR RNA, yeah, ribosomal, the gene on the mitochondrial, yeah, it's one of the 3 or 4 what they call barcodes.
Right.
There's cytochrome B and CO1 and CO2 and 12SR RNA. So,
And you're
but I only got 220 bases. This technique doesn't give you a very long sequence.
Oh. And you're pretty well guaranteed to identify some human DNA.
Oh yeah.
And
Well, I use the mammal primer, and then I go through and search my list of sequences for every little site against the database and find the human ones. Actually, I use the human reference to compare to. And then I go from there and I end up with a chart that has for a sample, all the mutations and or sequencing errors. And I have a way of separating out the sequencing errors. I used my own urine, and I have the same sequence in that range as the Cambridge reference sequence. So I have no mutation. So even if I contaminated the sample, any mutations from the reference are going to be real, not me.
Breaking news, Dr. Hart's new study shows that he is related to all the Sasquatch in Texas.
Yeah, that's right. It's a matter of how close. My detractors would say very close.
Yeah. There's a guy out there by the name of Mark Zaski who claims that he has blood of the Sasquatch in him and that he's related to a Sasquatch. I want to try getting his DNA sample too.
Well, I'd love to do that. Now, it's interesting that Sykes, when he did his study of Cheddar Man, which is a Cro-Magnon type man, I think over 10,000 years old in England, and he took DNA from a large sample of people in that neighborhood. It's the name of the place is Cheddar, and he found one person in this whole town that had the same DNA as Cheddar Man.
Wow.
Exact match. The mitochondrial, anyway.
That's pretty cool.
Which is interesting.
Yeah.
People don't move around as much as they you think they do.
I guess that guy is what you call autochthonous.
Is that the word?
I think that's the word. That's one of those 50-cent words.
That's more than 50 cents. I've never heard it. I'll have to look it up.
Kaiju Ninja 1985 said, how many DNA scientists are involved with the Bigfoot DNA analysis? Why aren't there more? I think you could probably count them on one hand.
Yes.
Not really.
Todd Dissatel has looked at it a little bit.
Right.
A whole lot of scientists would be into it if there were a body on file.
Right.
You know,
Right.
If there was a specimen on file, then there'd be a whole lot of people looking into it.
Yeah. Well, that's one of my next things. I have a detector that uses radiation, and it can detect people or human remains. And so I'm thinking it might detect Sasquatch remains. So I'm going to try to find some real samples. I mean, bones or something.
Dr. Hart, are you getting into radiometrics?
Radiometric? Well,
radiometrics. Okay.
This uses very long wavelength radiation, and it hits the sample and comes back, and the interference pattern that's generated can be detected with a simple antenna.
Yep.
And so you get a line between the source and the sample, and then you can triangulate with another one, or if you follow that line, when you get to the sample, you get some unique antenna movements. So I've gone out to cemeteries. I find every single body there shows up, including kids, little kids that died over 100 years ago.
That is
And
cool.
so I'm working on this. And gosh, this would be great if I could find just one bone, you know.
Right.
Other thing is LIDAR, which is, again, it's a laser technique that sees through trees. And they map a whole area. And like in the Mayan Peninsula, they found new pyramids, you know, down there.
Right. And in the Amazon.
Yeah, in the Amazon, you're all covered with trees. So something like Google Earth won't help you. But I have found some very unique structures in this Ouachita area. And I think if this technique were applied and some new data obtained, you may find places where Sasquatch seek refuge, such as caves or shelters or something. I just can't think they sit out in the open in the winter, you know?
Be the same way. Yeah.
Find any structures that you think might represent grave sites?
I have not, but
burial.
I'm going to look. I mean, if this detector of mine works, I might find something. Yeah.
Of course, then.
No, I haven't up to this point.
That's a whole big can of worms about Native American possible remains. That's one big source of what am I trying to say here? One reason that there's not more research going on is the assumption is that if you find old bones and they're older than historic period, then it must be Native American, and that's off limits.
Yes. And so now you got the government wanting you to, of course, reduce them and contact the tribe and try to get them reinterred.
Right, and by all means, do not test them or figure out what it is.
Yeah.
Kaidu Ninja says, how much DNA can you get from a footprint? Obviously, it depends on the environment, of course. Yes. That's the thing about the eDNA, Kaiju Ninja.
Yeah, that would be eDNA, the soil underneath, or even on snow, a snow footprint could yield some, even if there's no hair, which would be ideal, but just walking sloughs off cells, which are going through a stream, you know, another thing. You get these cells sloughing off, and that's where the DNA is.
I think I'm right in saying that the one of the best samples you could get would be a scat sample, because it would have cells that were shed from the colon.
Yeah. Oh, yeah.
But, you know.
That would be better than hair, or probably better than soil. And then you'd find out what he eats as well.
Right.
And it would also have all the DNA from the intestinal flora and.
Yeah, that's what I'm saying. What he eats. Right. Absolutely.
So
that would be a great find.
This whole eDNA thing, especially in criminal investigations now, is like, oh, you can leave a fingerprint behind, but now they can actually get skin cells off that fingerprint and DNA test it. So the heck with actually matching up the fingerprint, you're going to get DNA off that fingerprint now, and you're going to get matched that way.
Yeah.
Or your cigarette butts or anything like that.
Right.
Yeah, of course you'd have to collect one from the suspect.
Right.
Whereas with the fingerprints, of course, you may have him in the crime database too. So,
you know, that's kind of odd as they tie in this stuff to technology. You know, you got your phones now where you can stick your thumbprint on there, and then when it gets to where you have to lick your phone before you make a phone call or send a text, I'm drawing the line. I'm not doing it. No, sir.
We're headed that way, believe me.
Well, yeah,
I think so.
My crazy cousin Mario, you know, he terrible guy. He used to, he used to go to bars a lot, and then he would call up all his girlfriends on his phone. So he's actually got a phone now that he has to blow into, and if he's over the limit, it won't let him make a phone call.
Oh my God. Yeah, these cell phones are something else, and they're your whole life.
It's amazing how technology has brought us so far forward in a lot of different aspects. Um, I'm a very, as people know, I'm a very big proponent of AI in certain aspects because I think ethical uses of AI is a wonderful thing. It has enriched this program tremendously, both graphically and information wise. But we have the advancements of DNA collection. We have the advancements of cameras. I mean, I don't use a video camera anymore. I have an extra Galaxy Droid phone with a really good camera on it. I can slap portable microphones on the thing, and it sounds just as good, and it takes just as good quality video as a video camera did. Heck, it even takes 4K video. And so the technologies are getting so much better. But one thing that people will say, well, so many people have these phones. That why aren't we getting more pictures of or videos of Sasquatch? And I explained that in my latest book, but it is so difficult. I see a Bigfoot. Let me get my phone out. Let me pull it out. Let me make sure it's turned on. Let me get my thumbprint on there. Now I got that up. Now let me call up my phone app. Now, oh, it's gone.
Yeah.
So, and that's if I'm calm, cool, and collected. If I'm like,
well, when you reach for your phone, it hit the ground because you're excited.
Right.
Okay, Sasquatch Wizard Adirondack said, so do any of these samples still exist? Great question. And if so, could they now be given to Darby Orcutt's project at North Carolina University? Now, don't they normally, isn't the sample normally destroyed when the DNA is collected from it?
It depends on how big it is.
Well, that's right, yeah.
But here's the $1 million question. Do you think Dr. Ketchum would release that to Dr. Darby Orcutt?
No.
Definitely not.
No.
But yeah, normally, your buccal swab or a hair sample would be totally consumed unless they had enough DNA extracted where they could hold some back. If it's a big enough sample, you get enough DNA, you might not need to sequence all of it. You could save some.
The steak sample, that would have been.
There might be the possibility that some of the submitters did not submit their entire sample.
Yes,
that they only took a piece of their sample and submitted.
That's very likely.
Well, with Dr. Ketchum's study, didn't she find that there were some of this human factors in the sample that was considered the steak, but also Dr. Brian Sykes did he found that to be bear.
Correct. Yeah. And actually, we talked about that a little bit earlier. And actually, there was four scientists that looked at that steak, and Dr. Ketchum was the only one that came out and said that was a novel hominin, or was about to. Until
that's right. That's right.
The other three all tested it positive for bear.
And then I took her own data and proved it was a bear. So
right.
She was just had to be a misinterpretation. Whether it was intentional or unintentional is the question. Yeah.
Well, I think a lot of it's confirmation bias, and let me mold this evidence around my theory rather than the other way around. Because she's truly not acting like a scientist in her responses to other scientists. She's acting like a petulant, person who comes up with their own Bigfoot evidence, and despite everybody saying that's not a Bigfoot print, or that's not that's a shadow in a tree, that's a blob squatch, it's a shadow, they're going to hold on to that and fight for it because they have so much confirmation bias in their head that they're not listening to reason, and that's one of her shortcomings is she's not listening to reason. If you acted like a scientist rather than a petulant person who found Bigfoot evidence that refuses or alleged Bigfoot evidence, I find this no different than a guy sitting there arguing with Dr. Jeff Meldrum last week on Facebook. Dr. Meldrum tell this guy found a foot track, cast it, and Dr. Meldrum looked at it and said, "That's not a Bigfoot print for the following reasons. Look where the foot flexibility is. It's too far, north of the midline to be a mid-tarsal break, so therefore it's not a Sasquatch, yada yada." And this guy was insisting that Dr. Meldrum was wrong. Oh, I've only been a researcher for 35 years.
Okay.
Again, Dr. Ketchum is not acting like a scientist. She's acting like a Bigfoot researcher who found some evidence and is insisting on the validity of their own found evidence. And that's what happens. That's what happens when you create a study where everything is yours, right? That's what happens. Now, all the results are mine. Well, no, maybe your sample, but all the results are mine. I own the results. I'll be the arbiter of those. You see what happens? It corrupts the mind, and it gives you bias. It gives you that power. So, of course, I want this to be Bigfoot because I want this to be this benchmark of a study. And then you start convincing yourself that this is Bigfoot. Oh, and then people start saying, "Oh, you're just jealous." It's just a bad cacophony of failures. That's it's like falling down a flight of stairs. Is that one thing is leading to the other? If she had just remained neutral like Dr. Orcutt, then it wouldn't matter. It is what it is. But she had an agenda.
Well, finally, the trackway says, "What's the thoughts on the DNA sample that was collected in Europe?" And I don't know anything about that one.
What kind of sample was that? I don't know either.
Yeah,
I haven't heard of it either.
No. That's interesting. If you got any more information on that trackway, could you just drop it in the chat there, bud? I'd like to check that out.
Finding a Trackway said, "What is Dr. Orcutt's window of completion?" No idea. You'd have to probably ask Dr. Orcutt that one.
I don't think he knows, frankly.
No, I think it's kind of this open.
Depends on a lot of unknowns.
Yeah, I don't think there's been a date set. It's just like whatever comes in, we'll see.
Yeah.
Yeah, I think we got that one. I think
there would be more. There would be more. We just need a body.
Everybody would want a piece or a hair or a skin sample or something if we just had a body.
I know that this question is like, how did all these hair samples? There was no vetting process. That part of the DNA process is a vetting process. So to understand DNA, they're gonna, it should show that in 3 of the 4 scientists that looked at the Smeha sample, it was all vetted to actually as a bear rather than ruling out. But then again, to rule out an indigenous mammal, if a Sasquatch existed, it would be an indigenous mammal too.
Well, the best simple vetting process for hair is a microscope, because different animals have very different looking hair under the microscope. And Melba Ketchum had somebody look at some of them, and I looked at the same one, and it's a bear. I mean, I looked at her picture, and it matches bear just perfectly, and bear is very unique looking. And I don't understand how he didn't come up with that. And I doubt that she did this on all the samples by some competent person, because there are some the FBI uses this, and they've written papers that are online available, and it helps you get started. And I bought myself a microscope for, it was like $300. That's plenty good for this. They're not that expensive. Any Bigfoot group of investigators that ought to have somebody with a microscope. Before you spend money on DNA, take a look at it.
And the beautiful thing about it too is, you can plug them right into your computer and take pictures of them.
Oh yeah,
Yeah,
all these mine. So I can't look through one of these microscope things. I have a lot of trouble visually with that, and it all shows up on a computer screen. And then you can take pictures and send them to people too.
Yeah, you can get
It's
opinions.
simple. It's cheap. I just don't know why more people don't do it. Cindy, can't think of her last, Dosen.
Yeah.
In British Columbia is really good at this. She's much more experienced at it than I am. She's been doing it longer, and she will write you a full report for nothing.
Trackway said that DNA sample was the teenager that collected it in England.
Oh, Daniel.
Oh, now that sounds a little more familiar.
Yes, Daniel. I forget his last name.
And by the way, they finally verified that there are some Black Panthers running around in England from DNA.
Nice. Yeah, I saw that. That is.
And awesome. I forget it was hare or scat or whatever, but there are at least one of them.
Very cool.
People have been seeing things that they thought were, but.
So we got a couple of more questions, and then we got time for 3 more questions, because the last one is going to open up a can of worms. So first we'll do the first. What are my thoughts on drones? I think drones are a wonderful tool. Again, any video, whether it be thermal or not, is not going to prove this mystery, solve this mystery. There will be somebody there in science that will say it's a hoax, it's a fake, it's a guy in a costume. So unfortunately, we need either a DNA sample or we need a specimen. And,
glue a bunch of fish hooks on your drone and then crash it into a Bigfoot if you see one. That'll work.
Get some blood.
We'll get a sample somewhere.
But drone's a wonderful tool. No doubt. As is a FLIR. It's a wonderful tool. It's not going to prove a Sasquatch exists. No matter how good the FLIR is, there'll be somebody to say that doesn't prove anything. And unless you can put your hands on it, and that includes a DNA sample. You can put your hands on a DNA sample. Science is not going to accept it. Let's face that. I mean, you can do a track, and people can get their hands on that, but that obviously hasn't proven anything either. So, John, Sasquatch Visitor Adirondacks asks, Dr. Harder, is the new system you're working with called GPR, ground penetrating radar?
Oh, I think this is for Dr. Hart rather than me.
Right, Dr. Hart. Yeah.
Oh, okay. No, it isn't. It operates above. Well, it sort of operates in the ground, but it's not radar. It's a much longer wavelength than radar. But
yeah, LiDAR, right?
Yeah, well, not LiDAR either. It's even longer. The wavelength can be up to and even over 1 kilometer long.
Wow.
So it's in what they call the acoustic frequency range. If the same frequency were sound, you would hear it, but it's light instead.
Sounds like the same kind of system they've been using on Skinwalker Ranch.
Oh, really? I don't watch that program.
Yeah, they're using a lot of different technologies to see what's under the ground there.
Yeah,
and they have something that's they call the ground penetrating radar, but it actually can go down like 1 kilometer underground and above as well. So
right,
sounds a lot. Okay,
it operates in a different way. It is radiation, and it gets similar results, but the science of it is different.
I'd love to take that thing into a place like If you think about it, there was this old story, I forget where in Texas, but there was supposed to be in the 1800s a crashed airship. They called them airships at the time instead of UFOs.
Oh
And
yeah, yeah.
supposedly they took an occupant and they buried him in a cemetery, but they don't know
Oh.
where in the cemetery he was buried.
Aurora,
I-
Texas. Yeah.
There you go. Chris knows.
Aurora, Texas.
In a well. I think they
Yeah.
put the debris down a well and sealed the well.
Yeah, it's great. It's a great story. I mean, that's the fun thing about doing this type of stuff. You can go down rabbit holes all day long. And speaking of rabbit holes, David Parker came up with this one. Some believe that Sasquatch or the Igigi, who were the workers for the Anunnaki, if they also created us, could that be the source of
Probably mitochondrial. Yeah.
mitochondrial DNA? And do we think that as human code, and what we think is human could be Anunnaki? I think that is complete science fiction.
Well, no, there's a lot of documentaries out there that talk about stuff like this, and I could see going down that well.
But it's like I said, it's a rabbit hole, and there's a lot of documentaries that talk a good game, but when you start parsing the fine information, like anything, you will find inaccuracies.
Well,
And I would like to see any scientific report on that so I can take a look at it and parse it for its inaccuracies or its flaws.
And if I understand this at all, I think if the Anunnaki did this genetic engineering and modified humans or modified the precursors of humans or something like that, all of that would be contained in the nuclear DNA, and it probably wouldn't be at all detectable through the mitochondria. And the mitochondria is what most field samples are working with, especially with ancient DNA. It's usually mitochondrial DNA. So it's, even if all of that was true, I don't know how you would identify it using DNA technology. I mean, I just don't know. It could, if all of that was true, I don't know how you would find the proof of it in the DNA.
Steve, the Curious Cryptid says, that would tie it well with Melba's Nephilim conjecture.
Nephilim, man.
The Nephilim conjecture.
Nephilim. Yeah,
Cool story, bro.
yeah. More cans of worms.
Yeah.
Well, anyway, we are getting towards the end of the show, and
No.
yeah, how quick it goes by.
Well, I've just really been interested with Professor Harder and Dr. Hart talking about this stuff. It's just most of it does go a little bit high up here over my head, but I can latch on to parts of it. There's parts of me is catching this stuff, and especially the common sense parts.
See, and again, as far as the last argument goes about the Igigi and the Anunnaki, and here we are trying to bring another mystery into the Sasquatch mystery, similar to the way we bring UFOs and other stuff that we've tried to, other people, these are a lot of things that are of conspiracy theory. And like I said, this is a science, fact-based show, investigatory, principle-based show. So unless you show me something groundbreaking, I'm sorry, that's not going to be accepted by me. It's not something we're definitely going to espouse. It's something we'll look at, but right now I haven't seen anything other than tall tales with no science to back it up whatsoever. I have not seen one scientific paper on the Anunnaki. So until that day happens, I haven't seen one scientific paper on the Nephilim or the Igigi for that matter. So until that happens, it's nonsensical to me.
a specimen would be very helpful for those also.
Right. Exactly. So, anyway, gentlemen, I want to thank Dr. Hart and Professor Harder for coming on tonight. It's been a great discussion. It's been great having you guys on, clarifying the points, trying to bring it down to our level. I've tried my best to do that. Hopefully I did some of that. I think tonight was one of the better ones that we can say and break down what was exactly wrong with that study. And it's nothing personal, and that's something I always wanted to say. It's nothing personal against Dr. Ketchum. I mean, I have my reasons why behaviorally she's doing what she's doing, but it's still nothing personal. I do not dislike the woman. I do not hate the woman. I'm sorry, your study is just not correct, and that's the bottom line. Scientifically, it's not correct. From an investigator standpoint, I wouldn't sit by it. So, but gentlemen, thank you so much for having on. I'll give you guys a couple of last words. So I'll start with Professor Harder, and we'll wrap up with Dr. Hart. Go ahead.
Well, thank you for having me on. I really appreciate it. It's my honor, especially be in the panel with Dr. Hart. I'm really indebted to Dr. Hart for his work that helped me, convinced me that I had something else that I could say about it. Anyway, thanks.
Thanks for having me on. I really appreciate it. This is definitely a great group to be talking to, and y'all have a lot to contribute that I didn't know about either. And so best wishes to y'all, and let's go out there and find that sample or something, or specimen, and solve this problem.
Yeah.
Amen. And gentlemen, again, it's been my honor to have you on the show. I look up to you guys very much so. You guys are some of the pioneers in the top shelf. All right, Mr. Bennett, take it.
Once again, thank you, Dr. Haskell Hart and Professor Kevin Harder. We appreciate it. Always a great time having you guys on. Very educational. Man, thank you so much.
Thank you, Chris.
All our people over there in chat, all our listeners out on the podcast. You guys come by the YouTube channel, check it out. We appreciate you guys. I always say this, but I always mean it. You're the greatest audience we could ever hope for. And you make this channel possible. And if it's your first time watching, you're on the channel tonight, hit that subscribe button, punch the like, share. Sharing is caring. We appreciate it. And we'll see you guys next time.
And on behalf of me and everybody here, again, thank you to our guest, Professor Kevin L. Harder and Dr. Haskell V. Hart. Thank you guys for coming on again tonight. And of course, we'll be back here Friday night, 9 p.m. Eastern, with an all-new Bigfoot Friday night, where we're going to be talking about the Bigfoot basics. Let's get back to the basics, guys. We've got a lot of new information to break out, and let's shape up and get ready for the fall squatching season, which for a lot of us is our busy time. And that's just right around the corner, I know. In just a mere month and a half, I'll be out there with a group of you YouTubers from this very channel. Again, we have the best audience in the world, but a bunch of you guys will be out there in the woods with me right after the Sasquatch Calling Festival in Whitehall, New York. So looking forward to the stomp in the woods with you guys. Anyway, we'll catch you all Friday night. Until then, stay safe, stay healthy, God bless, and of course, keep on squatching. We'll see you all Friday night.
We thank you for being here with us on Squatch-D TV. If you haven't taken the time yet, please hit that like button and subscribe to the channel. And oh yes, hit that notification bell too. We don't want you to miss anything. If you really like our content, you should consider becoming a member. And it's really inexpensive and a great way to support not only the show, but honest Bigfoot research. Everyone have a great week. Be safe and God bless. We will see you all here next time on Squatch-D TV. Keep on squatching.